I'm happy to introduce our next trainee speaker, Morgan Pate. She was one of my superstars here right at UNC Pembroke. Working in my lab, graduating summa cum laude chemistry. So another chemist. 5 papers in my lab, I believe, as an undergraduate. Very glad she decided to stay in the North Carolina family of ADRC. She chose Wake Forest and very recently completed her PhD in integrative physiology and pharmacology. So this may be her first invited talk as Dr. Pate. And no pushing and shoving, the line will form over here for postdoctoral offers.
I want to thank everyone for inviting me to speak. I'm very excited to be back at UNCP and to get to share some of my dissertation work. So today I'll be talking about in vivo brain EVISF and their proteomes are sex-dependently modulated by AD-related pathology. Okay, so as all of us are pretty familiar with here, that Alzheimer's disease is characterized by extracellular amyloid beta plaques and intracellular tau neurofibrillary tangles, and also extensive neurodegeneration, which as you can see here in this diagram is that healthy brain versus one with severe Alzheimer's disease. And I think we've had several other speakers who have talked about this. And as it's already been mentioned, that new biomarkers for Alzheimer's disease are really imperative for the field to be able to detect Alzheimer's earlier and also to understand more about the disease pathology. And this is where extracellular vesicles come in. Now, extracellular vesicles are released from all cell types, and they can be found in all fluids in the body. And there are different types of EVs, but still the field is pretty new, and it's really hard in vivo to separate the different types. But I'm just going to briefly mention that there are a few different types. You have apoptotic bodies, which signal cell death, and you also have microvesicles, which bud off from the plasma membrane. And these are considered somewhat larger extracellular vesicles. Then you also have exosomes, which are derived from the endosomal pathway and are a smaller sized EV. And what makes EVs interesting is that they can carry different proteins and contents on their surface, making them interesting biomarkers, but also they carry different cargo inside, so different proteins, metabolites, etc., which makes them of interest in how they're involved with pathology and to be able to be used as biomarkers. So traditionally in the field, especially in the context of Alzheimer's disease, if you're wanting to look at EVs, and there's been different studies that are looking at EVs as AD biomarkers, they're looking in the blood or the CSF to see how they change with AD pathogenesis. So that's if you're looking in vivo, you can only look in blood or CSF for the EVs. If you're wanting to know, okay, what's going on with an EV directly in the brain, that can only be done in postmortem brain tissue of humans, monkeys, mice, etc. And this is where my dissertation work came in. We're like, okay, but what's going on in a live, intact brain with extracellular vesicles in AD? So I use an established technique in vivo microdialysis, which is rather complicated, but basically you can infuse artificial CSF into the brain of a mouse and then collect out the brain's interstitial fluid. And this is commonly used to look at levels of amyloid beta and tau in the brain. And I was the first person to ever look at, could you look at extracellular vesicles in the interstitial fluid? So what's nice about this technique is that it can be performed in a live, unanesthetized mouse. Also, we can look at a specific brain region that's vulnerable to AD. In my study, I looked at the hippocampus. Also, this technique allows us to access the extracellular pool in the brain to be able to look in the interstitial fluid to look at cellular cooperation and heterogeneity. Because as we know, there's all these different brain cell types in the brain that are all playing a role in this pathology. And finally, I should mention that proteostasis is dysregulated in Alzheimer's disease. And EVs that are being looked at in the periphery, in the blood or CSF, may not actually be brain-derived. So we don't know if EVs from the brain are really reaching the peripheral pools. So we need better biomarkers to say truly whether EVs are directly coming from the brain. So as it's already been discussed today, we know that humans get this pathology of the extracellular plaques and the tau tangles. I chose to look in this mouse model, the APP-PS1, that they get amyloid plaques similar to what is seen in Alzheimer's disease. So as you can see here in this example, at 3 months, these mice do not have plaques, but by 9 months, they have extensive amyloid plaque pathology in their brains. And what's interesting about this model is that the females get more amyloid plaques in their brain. They get them before the males do. Both males and females get them, but there's more pathology in the females. It should be noted that this mouse model does not get tau tangles, so it's not a perfect model of Alzheimer's disease, but it does allow us to focus on one pathology at a time. So for my project, I looked in wild-type mice at 3 and 9 months of age, and then in the APP-PS1 mice at 3 and 9 months of age, again, before plaques are present, and then once plaques are present. And we also looked in male and female mice. And what was interesting that we found when we were able to look at these EVs that are found in the alive brain is that in male mice, the number of EVs increased with age. Then the number decreased when plaques were present. So you started seeing it decreasing with the presence of plaque pathology. However, there was no change in female mice. And this was just the beginning of us starting to notice these sex differences in EVs from the brain. Finally, we also looked at EV size, which sometimes changes in different diseases, but we saw no change in our study. I then compared the 4 groups that I looked at. We did proteomics and to look at the different proteins found in our EVs. And when I compared the 4 groups, I found 20 proteins specific to 3-month wild type, 2 specific to 9-month wild type, and then 5 that were specific to the 9-month APP-PS1 mice when plaques were present. There were no proteins specific to the 3-month APP-PS1, which we thought was interesting, but what I want to point out here is that these 5 proteins here that were found in our AD model mice, that we're looking at them as potential new AD-specific peripheral biomarkers for EVs, so we're studying this further. I also want to mention that these 168 proteins that were shared between all 4 groups could potentially reveal to us new brain EV-specific biomarkers, which, as I talked about at the very beginning, it's hard to say of EVs we're seeing in the blood or CSF whether they're really coming from the brain, and so we may be able to develop new biomarkers using those shared ones. When we looked at our EVs further, we found that the protein concentration within the EVs, so the amount of protein within the EVs, increased with age regardless of whether there was plaque pathology present or not. However, the protein diversity decreased. And what I mean by that is when I looked at all the different proteins that were found in our EVs, there was less variety with age and with genotype and with pathology. So another way that I showed this is I compared each of the groups to one another, and then basically to summarize all those results is that there was a decrease in protein diversity with age, genotype, and pathology that we were detecting in these EVs. Then we also looked at pathway analysis of the proteins, and what I found was when I compared the 4 different groups to one another is that there were proteostasis and inflammation pathways that were modulated in the APP-PS1. So if you see here at 3 months and at 9 months, they have in blue is pathways related to proteostasis, and red pathways related to inflammation. And so there were fewer proteostasis top 10 result pathways in the APP-PS1 mice and more inflammation-related pathways. So then we wanted to know, okay, we're seeing these differences between these groups in these EVs directly from the brain. We wanted to know what cell types these EVs might be arising from, where they might be coming from. So I looked at several different cell types in my study, but what I want to focus on today is the microglia results, and I correlated those with A-beta pathology in the mice. So to take a quick step back and to follow up kind of with our last talk is that there are, when you have plaques in the brain, which is here in blue, that you'll see this inflammatory response where microglia and astrocytes will surround the plaques. So we know that this happens, and what we also saw in our mice in this study is that they do have more activated microglia when pathology is present, especially in the females. Moreover, when we looked at A-beta deposition in the brain, we found that as there was more A-beta deposition, there was also more activated microglia. We saw this in the males with a similar trend also in the females. So then we wanted to look at this a little bit further, and so we looked at plaques here, X34, and we saw that, of course, at 9 months, these mice had plaques in their brain in the APP PS1 mice compared to wild type. Interestingly, when we looked at total microglia, so IVA1 staining, there's no difference across all the groups. However, when we looked at activated microglia, as I just mentioned, in the mice that get the plaque pathology, there is increased activated microglia. So then we looked at co-localization of all these markers, and what we found was that, not surprisingly, the microglia and the plaques all co-localized once there was plaques in the brain. But interestingly, at 3 months of age, there was already a greater increase response and co-localization in the 3-month males compared to the females. So males are already responding differently at 3 months. So to bring it back to the extracellular vesicles is that, let me go back just 1 second. Then when I was looking at the microglia EVs, what I found is that when plaques were present in the brain, there were more microglia-derived EVs in the brain. And this was only seen in males and not in females. So when I correlated this to A-beta pathology, the way that I think about this is that in a healthy brain, you have increased neuronal activity, healthy neuronal activity, you have greater levels of ISF-A-beta, but once there's plaques in the brain, the levels of A-beta, free A-beta in the interstitial fluid drop, similar to what is seen in CSF, because they're getting stuck in these sticky plaques. So when I did correlated A-beta pathology, as the A-beta levels drop, as they're getting stuck in the plaques, there are more microglia EVs. I saw this with correlated with A-beta 40, similar results with A-beta 42. And then similarly, when I looked at A-beta deposition, the more A-beta deposition there was, the more microglia EVs there were as well. And when I looked, correlated this with activated microglia, the more activated microglia there was, the more microglia EVs there were. And what was interesting is that all these correlations and results were only seen in the males. There were no correlations seen with the females. So we're like, why might this be? So we did some pathway analysis, and when we compared the males and females, what we found is that some AD-related pathways were appearing in the females that we weren't seeing in the males. And this was largely driven by the presence of clusterin and ApoE, which are genetic risk factors for Alzheimer's disease and may potentially be explaining why we're seeing these differences in males and females in our extracellular vesicles. So just as a quick summary, basically the takeaway for this is that hippocampal EVISF response to A-beta is sexually dimorphic and correlated with the microglia EV proteome. And with that, I would like to thank everybody in the Macaulay lab at Wake Forest, especially my mentor, Dr. Shannon Macaulay, and everyone who helped with this project, shown with the asterisks here. I had a lot of help with this project, which I'm really thankful for, and our awesome collaborators who helped us isolate and characterize the EVs, my thesis committee, and also all of our funding, including my F31 NRSA, which I got for my dissertation project. And with that, I will take any questions.
So, you know, in a lot of Alzheimer transgenic models, the females are more affected or more functionally defective than the males are.
Mm-hmm.
Can you speculate whether the EV changes in the males might have something to do with, cognitive behavior protection compared to females or anything related? Do you think the EVs would have something to do with the male-female differences you see in terms of function in these mice?
I think potentially yes, or the way that I think about it is that EVs can be used for as a clearance mechanism, and they can also signal. They just have lots of different things that they can do. And I think like in this case, in my study, I think that the responses that the EVs or the cells are responding potentially better in a protective way in the males, or they're at least responding, how should I say it? That maybe males and females are responding the same, but we missed it with the females. Like what we're seeing happened earlier in the females, and we just like missed it. So it would be great to go back and look at it 6 months, where I just looked at 3 and 9 months. So whether this is truly like a protective thing with the males, or they're just a little behind what happens with the females, would be nice to study.
I just want to congratulate you on having both male and female mice, because that's not something that everybody does. And if you didn't do that, you wouldn't have found that's a very interesting result. And so just as an encouragement to people, because female mice are just much easier to work with, and yeah, that's so good. And I did mention over optimizing females, right? Yeah.
Yes.
Talk about